1, 2. Welcome to Biology 1440. This will be over Chapter 3, what I call the Legos of life, the molecules that make up our bodies, basically, the molecules of life. And so, getting this to work will be step 1. Okay, here we go. Now, we're talking about organic molecules in this chapter, and organic molecules are molecules that contain carbon and hydrogen. That's what makes them organic, okay? And they have carbon and hydrogen backbones typically, and carbon and hydrogen have roughly equal electronegativity. That means they share electrons equally. So you'll get a carbon backbone like this with hydrogens attached, right? And they're forming nonpolar covalent bonds. That means that this molecule itself is typically nonpolar. Of course, we as living things are made of water, and so we have to do something about that because we can't have chemicals in our bodies that don't like water if we're going to be composed of those chemicals. So, oils are made up of carbon and hydrogen, and they are repelled by water because they're hydrophobic, because they're composed of nonpolar covalent bonds. But we can make carbon and hydrogen backbones react with water or mix with water by adding on functional groups. Functional groups are chemical groups that retain chemical properties no matter where they occur. So if I have a carbon backbone like this, and it's got hydrogens attached to it here and here and here and here, but then I stick an OH on it here, O and H share electrons unequally, and this is polar, and this will allow this part of the molecule to interact with water, okay? So a functional group. Here's an example. Now, once again, functional groups kind of bring to light that idea again that chemical structure and function are tied together. For example, these two molecules have real similar carbon backbones. That's what all these stop sign looking things are. But they have a couple of slightly different functional groups in slightly different places, which makes this one estrogen and this one testosterone, right? Here we see an OH down here, whereas over here we have an oxygen double bonded to that carbon backbone. There are 7 functional groups that are important for life, that are the most important for life. Hydroxyl groups, which will look like an OH. Carboxyl groups, which are an OH and a carbon, and then double bonded to an oxygen. That's carboxyl. This letter R stands for the rest of the molecule. That's what that stands for. Amino groups, phosphate groups. These I'm highlighting because they come up in this chapter a little bit, at least several of them do. Amino groups you find in amino acids. Phosphate groups you'll find in DNA and ATP and things like that. We also have carbonyl groups, which will just be a carbon double bonded to an oxygen, for example. We have sulfhydryl groups. Sulfhydryl groups, as we see there, has a sulfur in it. Or, let's see, what was the last one? The carbonyl sulfur. Anyway, we got them all. We got them all there. These are all the functional groups that you'll see a lot in chemistry, and we're not going to see maybe all of these in this chapter, but we'll see a couple of them, and I'll point them out as we go. So functional groups give organic chemicals unique properties, as well as the backbone itself. The backbone can vary in length, like here's ethane and propane. This one has 2 carbons, this one has 3, right? It can vary in terms of branching. So for example, notice there's 1, 2, 3, 4 carbons here. Here are 4 carbons as well, but here it branches. It's 3 this way and 1 up. So branching can change the chemical structure of the molecule. We can move where the double bond is, and that changes the chemical structure. So this is 1 butene and this is 2 butene. Also the presence of rings, like when I drew that, we talked about those stop signs before. This is what they look like when you draw all the carbons out. There can be double bonds, which is drawn like that, single bonds, hydrogens attached, etc. So the backbone, this is how that's how the backbone can vary. So variations in backbone. Once again, variations in the functional groups, so hydroxyl groups, carbonyl groups, carboxyl, amino, sulfonyl dihydral, or phosphate. Also, getting back to this branching idea, I want to show you here. We call these isomers. Isomers are when you have the same parts in a different structure, kind of like we saw in an example like we see here, where there's the same parts but in different orientation. So if you count the number of carbons and the number of hydrogens here, you'll find that they're the same, but they're just arranged differently. So an isomer is when you have the same parts, different structure. All right, I'm sorry that that's illegible, but that's what it is. And then that's an example of a structural isomer. A trans-cis isomer is when you have the same parts and they're almost put together the same way, but one might be a mirror image to the other, for example. Actually, let me rephrase that. You could have cis-trans isomers like this one, where here, whatever this is here, these functional groups or whatever, are side by side, and in this case, they're opposite one another. Cis-trans. Cis being the same side, trans, they're opposite. Enantiomers is a type of isomer, which is the mirror image version, where you have a mirror image of the molecule being slightly different. So isomers are once again same parts, different orientation. That definition really covers all three of these. And so just have a look at that and see if you can see the differences. You might not think that's a big deal, but that changes how your body interacts with these things. For example, there are drugs that in this form would not cause any effect on you, and in this form they might help you relieve pain or something, and they don't work if you give somebody the wrong version of the drug. All right, so throughout this chapter, I'm just going to reference the digestive system, just because I thought it was a good way to frame this chapter. Your digestive system, you'll learn more about in Bio 2, but the major organs that we're going to worry about being the stomach, where proteins are digested, and the small intestine, where a lot of other digestion occurs. So when you eat your food, you chew it up. Your saliva begins to break down carbohydrates. You lubricate the food, you swallow it. It hits your large intestine. It's broken down further. The proteins are denatured. So that means the proteins come apart. They go into your small intestine, where things are broken down more and absorbed into your blood. And then that food is passed throughout your blood to your body, where cellular respiration takes place, right? And so we're talking about the molecules that are being passed into your cells by your blood after you eat your Pop-Tart in the morning. And most of the molecules in this chapter we call macromolecules. Macromolecules. Why? Because they're big. And most of them are found on food labels. Fats, right? Carbohydrates, proteins. The one major category that I don't see on here would be nucleic acids. I guess people would freak out if you had your daily serving of DNA on your food label. But all of your food that comes from living things does indeed have DNA inside. In the next lecture, we'll start with fats or lipids, and we'll work our way through the major macromolecules found in living things.